Hi everybody. Thank you. My name's Karen. I'm a professor of geomicrobiology at the Open University in Milton Keynes. I am a microbiologist by background, so I have a BSc Honours in microbiology from Heriot-Watt University, and I have a PhD in microbiology as well. And I was lucky enough to do that at the University of Otago in New Zealand. Okay. So are we alone in the universe is probably one of the most fundamental questions of our time and is the driving force behind astrobiology. For thousands of years, peoples have looked up into the stars and wondered about the presence of life elsewhere and if other habitable planets exist. Although our understanding of the solar system is constantly evolving, no ambiguous evidence of life has yet been found elsewhere in the solar system. Therefore, it is key to look for life. If we want to look for life, we need to look for Earth. We just understand if, where, and what evidence can be found. To do this, we use environments on Earth. Particularly interest are extremophiles, microbes that live in extreme environments on Earth, which can be used as analogues, what we heard about before, for looking at potential life elsewhere. So in this talk, I'm going to introduce you to extremophiles, some of the most fascinating organisms on this Earth, and explain to you how these can give us insight for looking for life elsewhere in the solar system. So most extremophiles that we know are microorganisms. So these are small microscopic organisms which we can't see with just our eye. Now, estimations predict there's over 5 nanillion organisms on Earth at the present day, and that they're the major contribution to the world's biomass. And no two microorganisms are the same. There's huge variation between them. So for example, if we look at their morphology, we can see that there's variation. So we're showing this picture here. We can have coccoids, so round cells. We can have rod-shaped ones, so vibrio. We can even have some kind of spiral ones as well. But one thing all life has in common is that it has 3 key requirements. It needs liquid water, which is a solvent which is needed within the cell for cellular processes. It also needs key elements. So, for example, carbon, hydrogen, and oxygen make up organic molecules and kind of like the backbone of cells. We need elements such as phosphorus and sulfur. So phosphorus, again, is really important for DNA and also to ATP, which is a molecule which is used for like the currency of the cell. They also need an energy source as well, which I will talk to you in the next slide. Now, if you look at this figure here, you'll start seeing similar patterns to what we look for habitability beyond the Earth. So when we look for life beyond the Earth in environments which are potentially habitable, we look for finding evidence of water, we look for these bioessential elements and energy sources. So for examples of this, looking at habitable missions going forward, we have missions to the icy moons, where we know that there's subsurface oceans that exist. And some of these icy moons, so for example, Europa and Enceladus, we predict that there's some hydrothermic activity within these subsurface environments, which could produce some of these chemicals that are needed for life. More closer to home, we look for Mars as potential habitable environments as well. So looking at geological evidence, we predict that on early Mars, there were areas where large fluvial systems existed. And we know from what we know on Earth that microbes are really flexible. So for example, if we look at energy on Earth, a lot of organisms and life primary production is driven by light, so photosynthesis. So we look for trees and plants, they use energy from the sun to produce energy for growth. But that's not just the only example. So for example, if we look in deep subsurface environments where there's no light present, organisms can use chemical energy. These organisms are called chemotrophs, and they either can use inorganic chemistry, so we call them chemical eaters, or lithotrophs, so rock eating, so you can eat parts of rocks to get the energy for that, or they can use organic, so these are called organotrophs. Now, so as well as the key requirements that we have previously mentioned about water, key elements for life, and an energy source for growth, they also need to make sure that they have the physical chemical conditions as suitable as well. So if we look at this kind of complex diagram here, it sums up really nicely about what we know. So the green box in the middle is kind of familiar life to us as humans, where the conditions aren't that extreme. We've got blood, which is about pH 7, with temperatures, we're quite happy as humans, living, surviving about between 0 and just over 40 degrees. We're quite happy there. We're in this little bubble. But what microorganisms can do is they can kind of push this envelope. They can push some of these environmental extremes so that they can survive more extreme environments. So, for example, some of the first extremophiles we've looked at were in the Yellowstone National Park, where we have these hot springs where they have high temperatures and extremes of pH, so down to about pH 2 up to about just over pH 8. And we know from looking at the microbes that can live in there that there's actually habitable environments where microbes can live. And they're not the only examples. As you can see in this diagram here, we have some microbes that can live in acid environments, etc., and some in these cold environments. And what we do know that over the last century, that our understanding of the boundaries of life has evolved dramatically. And we've identified microbes in some of these environments where we initially thought were inhabitable. So this can be from anything from found in ice sheets to saturated salt pans and alkaline environments such as lakes, and also in human-made pollution environments as well. So we've got evidence that microbes can survive in nuclear containment sites and in high toxic chemical environments as well. And these general term are called extremophiles, but depending on the physical chemical parameter that they can survive in, they are specific to kind of different areas. So, for example, if we look at temperature, we have thermophiles and hyperthermophiles that can live at high temperatures, whereas at the other end of the spectrum, we can have cold-loving microbes. So, for example, the cryophiles that can live below 0. We have a very similar situation with pH, where we have high organisms that live in high and low pH again. So depending on the environments that they can survive, it depends on what we call these organisms. And for microbes to live in these extreme environments, I suppose to them it's their norm. For us, we wouldn't be able to survive in these environments, but they can. They've actually physiologically adapted to these conditions. So what they've done is they've changed their cell walls or their cell makeup so that they can survive in these conditions. So let's look at some of this in a bit more detail. So if we take, for example, the Helophilics, which, as I mentioned before, are kind of these salt-loving microbes that can live in high salt concentrations. So as we see here on the picture on the right, we see these kind of salt flats with these really hypersaline lakes where we know that microorganisms can live. Now, for a non-extremophile, non-helophilic organism to live in these environments, it would be really difficult for them to live. Because what you have is you've got a really salty environment, and what happens is that the cell membrane that surrounds the cell is impermeable, which allows water to pass across these membranes quite easily. So what happens if you didn't have an organism that could survive in these conditions, the water would leak out of the cell, causing dehydration to occur. But what we find in Helophiles is that all helophilic organisms have this kind of equal osmotic potential across the membrane, which means that water doesn't leak out of the cells in these hypersaline environments. Now, there's two ways that they can do that, depending on the kind of organisms that we have. We have organisms that can produce that what they do is they take in ions into the cell that make a really saline environment within the cell as well. So there's kind of an equilibrium across the cell membrane. This is dependent on the organism producing having cellular processes such as enzymes that can survive in these conditions. Or an alternative is that what happens is the cell produces these organic solutes, which produces a equilibrium across the cell, but without having to have that added complication of adaptation to these salty environments. So if we look at extremophiles from an evolutionary perspective on Earth, what we see is that they're not specific. They can be found throughout the tree of life in the domains of bacteria, and particularly in the domain of bacteria and archaea. There's a whole variety of them. And what we find that the deeply rooted ones, the ones that are most closely related to the last common ancestor, are these kind of these thermophilic organisms that we see grouped together. And what we predict from this is that some of the environments where life originated on early Earth was from these kind of hyper thermophilic environments, which could be a hydrothermic vent in the subsurface, similar to the subsurface ocean, or on the side of a volcanic environment. So what's important, I want to take home message from this, that extremophiles are not just important from looking from an astrobiology perspective, but they're also really important from looking at an evolutionary perspective, because they have this kind of flexible genome, which has allowed them to adapt through time. And as well as having these kind of extremophiles, which I mentioned, we also have these kind of superheroes of the extremophile world, which can survive in more than one extreme. These are called polyextremophiles that can live in high pH, high temperature, and radiation. So there's a whole variety of organisms that can be found. And as time goes on, we're isolating more and more of these organisms. So what does this mean? So what we can do here is we look at Earth to kind of predict what we can look for habitability beyond the Earth. So if you look at this diagram here on the top left, where we look at Earth beings, they've been mapped in this paper where microorganisms are known to live in relationship to salinity, pressure, and pH and temperature. When we map that then against some of the potential environments that we know on other planets, celestial bodies, and icy moons, we can see that there's areas of potential overlap where these conditions can exist. However, we must be mindful that within this diagram, they don't include water activity and radiation, which are key parameters for looking at survivability. But it kind of gives you an idea that there are regions beyond the Earth where, if everything matched up, there could potentially be environments where life could exist. So let's look at an example of this. So a prime example is if we want to look at early Mars. So what we know about Mars is that the present day conditions is really cold, dry, heavily oxidizing, and exposed to high levels of radiation. Now, these conditions are deemed inhospitable to life, but if we look at geological and geomorphological data, we could predict that conditions on early Mars, the conditions they were a lot more clement than they are now. And data from MSL, from Gale Crater, and especially this pivotal paper here on the right, have suggested that the key ingredients for life, so an energy source, water, and bioessential elements, would have existed within these Gale Crater environment, suggesting that once upon a time, the environments on Mars was habitable. And what we can do on Earth is that we study analogues that are similar to these kind of conditions to get an understanding. So very similar to what Sam was saying before, my group and microbiologists look at these analog environments to see the feasibility of habitability within these environments, and using that information to refer us about habitability elsewhere and the biosignatures they can produce. So a prime example of that is some work that one of my postdocs was doing, looking at the Helophilics Island in the high Arctic, which is a really salty, cold permafrost environment, which is rich in sulfur salts, and has been used as an analogue for looking at the Noachian to Hesperian transition on early Mars. So as part of this study, we went there, my colleague Mark Papel went there, and he collected samples from this site, and we kind of started to look at the community and the geochemistry within these environments to kind of infer about how life could potentially have existed within the early Mars conditions. So from a microbiology perspective, we were really interested in looking at what kind of microorganisms can live in there. So going back to what I was saying about the energy sources, what we were able to determine was that a lot of organisms within these environments were dependent on particularly sulfur to produce energy. So it suggests that the primary production within these environments would be dependent on sulfur and sulfur metabolism existing. And what we can do is we can use that kind of understanding of what organisms can live in these analog environments and use that to start predicting what could happen on Mars. So using Mars data, so this was taken from MSL data, we can start to use thermochemical modeling to kind of predict the water chemistry and what redox reactions can occur to allow microbial organisms to grow and produce energy. And from this, what we can start to do is look at what kind of ecosystems could potentially exist, and also predicting kind of biomass numbers that could be produced and supported by these early Martian environments. And this is really important for emission data because we need to be able to understand the limitations of our implementation and also, the cell numbers that we're looking for. And from this information, we can start looking at biosignatures. So these are molecules that can be used as evidence of life to determine the presence of life. So using analog environments, a key part of it then is to look at how these biosignatures or molecules would be able to survive when exposed to extreme environments associated with these extraterrestrial environments. So we've been involved in a number of exposed experiments on the International Space Station, where what we've done is we've exposed microbes and biosignatures to the conditions of low Earth orbit. And we can use this also to simulate the surface conditions on Mars to look how biosignatures would actually degrade and break down in these environments. And this is really important if we want to start understanding how we can detect these with instrumentation that we either have on the surface of Mars at the moment. And the aim of this, the most recent experiment that we've been doing is Biosign, which is a paper that will be coming out soon, is what we've done is that we've taken some biosignatures that we know, some molecules that we know that are key for life, mixed them with some analog Martian simulant regolith, exposed them to the conditions in low Earth orbit for a couple of years, and brought them back. And then using Raman spectrometer, we've been able to look at how these biosignatures degrade over time and if they are actually being able to use them as a biosignature for these missions. And obviously, we can't always go to space to do these experiments. So we also have a number of environmental simulation facilities at universities where we can simulate these conditions as well. So I think for me, the conclusion is extremophiles are really exciting and really diverse organisms that we can use. They're kind of like the superheroes in the microbial world, and they can adapt and survive in some of the most diverse environments that we know. And if we are going to find evidence of life beyond the Earth, understanding the physiology and how these organisms adapt are really important to this quest. Thank you.
Thank you, Karen. Again, we've only got time for just one question. So I would like to ask, could you tell us a bit more about some of your upcoming research that you're going to be working on?
Yeah, so the key sites that we're looking at the moment are, we're looking at Botswana. So we're working in the salt pans in Botswana, which we were supposed to be going in March, but we'll wait and see if that happens. And what we're doing is we're using that environment as an analog, that paleo lake environment as an analog for kind of Gale Crater. So we're working there with local universities where we're looking at the microbial ecology and looking at biosignatures there. We're also doing work in Ethiopia, where we're looking at some of the salt lakes there as well. So we're doing a lot of work in African countries for analogues, and a key part of that I'm really strongly support is working with the local communities in that area as well. We've really been supported. We go in the field together. We analyze data and share samples, and I think that's really important with some of this analog work. Is that we ensure that we go and we do it in an equal and fair manner with the local communities as well.
Thanks. That's really great. It's really great.
Can I just say we have a number of PhDs that are on offer. So if anyone's interested, please go to the AstrobiologyOU website. I think we've got 8 PhDs on offer at the moment. So please go and check them out if you're interested in doing any PhDs in this area. Thanks.